Hello students, welcome to Program Bachelor of Science 3rd year. The subject Botany, Semester 6, Course Code BOC 108, Course Title Cytogenetics and Plant Breeding. This is Unit 9, Methods of Crop Improvement. The module name is Role of Mutation and Polyploidy in Crop Improvement. I am Ms. Marina Janice Rodriguez, Assistant Professor in DCT's Dempi College of Arts and Science, Miramar, Goa. In this module, you will be learning about the role of mutation in crop improvement and the role of polyploidy in crop improvement. By the end of this module, you will be able to describe the methods of inducing mutation, handling mutant materials, use of mutant genes. You will be able to summarize the impact of mutant cultivars in plant breeding and also explain the role of polyploidy in crop improvement. Let us begin. Role of mutation in crop improvement. Mutation can produce many important qualities in plants. Therefore, it can be used in changing the genotypes and phenotypes of plants to produce the desired strains. Mutation breeding is a term which refers to the induction of mutation in plants with the help of mutagens and their utilization for the production of superior qualities in plants. In plants, mutation can be induced through various agents, what we call as mutagens. Now, mutagens can be physical or chemical. Physical mutagens can include radiations such as X-rays, UV rays, gamma rays. Physical mutagens can also be a change in temperature, that is heat shock treatment. Chemical mutagens can also be used to induce mutation in plants. Methods of inducing mutation. The first one, treatment of plant materials with physical mutagens. Plant parts such as seeds, bulbs, and rhizomes of perfectly produced plant varieties are irradiated with X-rays, gamma rays, UV rays under controlled conditions. Now, these controlled conditions can be the conditions of temperature, oxygen levels, humidity, and so on. The controlled and treated seeds are then germinated in petri dishes and the germination percentage is recorded. After that, the germinated seeds are transferred to experimental plots and the emergence of seedlings in the field is recorded. The particular radiation dose which reduces the germination to 50% is considered as lethal. Second method, treatment of plant materials with chemical mutagens. Chemical mutagens, when used to induce mutation, are usually used in a solution form. The mutation frequency of chemical mutagen depends on the dose. Now, the dose is calculated as concentration into time. The mutation frequency also depends on the temperature, the pH level, and some other factors. Let us see how it is done. The seeds are usually dried and pre-soaked in distilled water for a few hours because it has been observed that seeds which are not soaked do not give good results as compared to the seeds which are soaked in distilled water for a few hours before the treatment. These seeds are then placed in freshly prepared solutions of chemical mutagens. Chemical mutagens which are usually used are ethyl methane sulfonate, ethylene or methyl methane sulfonate. The treatment of plant materials are washed in running water for 2 to 3 hours and then placed in petri dishes for recording the germination percentage. The germinated seeds are finally transferred to experimental plots. Handling of mutant materials. Once we have induced the mutation, the population that is treated by the mutagens is designated as M1, the first generation of mutant variety. The plants of M1 generation are carefully studied for their morphology and reproductive behavior. because the primary effects of mutagens are usually seen in their color, shape, venation of leaves, branching or flowering patterns. The seeds of these abnormal plants in each treatment are collected separately. Usually 200 to 2000 seeds are collected. 10 seeds from each M1 plant will be enough for raising M2 generation. Selection of plants with desired characters can be made from the M2 population. 2 to 5 best possible plants for a particular desired character are selected from one row and are selved. These seeds obtained for such plants are grown to raise M3 generation. The use of mutant genes. Direct use. Now, when certain true breeding mutant lines with desired genes are established, then suitable breeding methods are employed to obtain desired genotypes. In autogamous species, mutants selected in M3 generation can be used directly as genotype if they breed true for a particular character. For example, Shabarbati sonara is a single gene mutant in wheat population which has been derived from Sonara 64 variety. In cross-pollinated population, there occurs a greater degree of heterozygosity. So the selected mutants should be allowed to inbreed for 6 or 7 generations. If they breed true after this inbreeding, then they can be used as mutant genotypes. Incorporation of mutant genes in self-fertilized crops. The desired genotype recombination after M3 generation can be obtained from autogamous species in the following manner. The first, the mutant is crossed with parental variety. Second, 2 or more mutants from the same parental variety are involved in pedigree program. Third, backcrossing mutant as donor parent with different variety as recipient parent. Incorporation of mutant genes in allogamous species. In cross-fertilized species, the desired genotype can be achieved through hybridization by mutants followed by inbreeding recurrent selection. Through mutation breeding in cross-pollinated crops is difficult, mutagenesis increases the variability to widen the range for selection. It can be used in developing male sterile lines. Genetic variability is a result of induced mutation by various mutagens which have contributed to modern plant breeding in the development of superior plant varieties. Several achievements in crop improvement through mutation breeding have resulted in improvement varieties that are directly used for commercial cultivation and also in new genetic stocks with improved characters with better combining ability of traits. These traits that we talk about may include resistance to pest diseases, enhanced nutritional quality, early maturity, drought and salt tolerance. The genetic stocks developed from mutations can have numerous applications in plant breeding. As we have seen, they can be used as donor parent in conventional plant breeding programs or as a parent in producing hybrid. Role of polyploidy in crop improvement. The polyploidy is a condition in which cells of an organism have more than 2 paired homologous sets of chromosomes. There are 2 types. Autopolyploidy, duplication of a single genome, and allopolyploidy, combination of 2 or more differentiated genomes. The first application, genome buffering and allele dosage. The presence of multiple genomes in polyploid plants would retard the response to natural or artificial selection due to mutation and recombination. Some traits may be buffered from mutation by genome redundancy even when underlying genes are mutated. Example, genome buffering for the waxy locus of potato which buffer the plant against mutant alleles. Polyploidy affects allelic dosage. When polyploid nucleus is formed, every gene in the genome becomes instantly duplicated, leading to novel dosage effects that may be important to phenotype. Second application, increased allelic diversity and heterozygosity. Having only a single genotype Autotetraploids do not have intergenome heterozygosity. However, it may be possible to combine pyramid blocks of genes containing diverse alleles into single polyploid line with the goal being to maximize allelic diversity. These are the examples of analytic breeding method for autopolyploid potato and increased heterozygosity in autotetraploid alfalfa. Progressive heterosis is maximum heterozygosity which was obtained after intermating double-cross progeny for one or more additional generations beyond the final F1 line. One important aspect of allelic diversity is that concept applies not only to coding regions but also to the regulatory sequences, which, like the coding regions, they control, experience independent evolutionary pressures while separated in diploid progenitors. Even when the desired allele has been chosen, there may be several strategies for optimizing the plant breeding effort. 1. Increase allelic dosage if desired transgressive segregation by homologous recombination. Selecting a single dominant allele for its phenotypic effects but relaxing dosage selection, thereby reducing the number of selected markers and their accompanying linkage drag. The third application is novel phenotype variation. Novel phenotypic variation is known to accompany polyploidization. Example, in Brassica, significant de novo variation was found for flowering time and for several life history traits. There are many different mechanisms that could give rise to these types of novel phenotypic variation and may provide selectional basis for crop improvement. Last one is polyploidization and genetic bottlenecks. While potential allelic diversity with polyploid genome is greater than the diploid genomes, often there exists higher levels of natural variation in related diploid species because the process of polyploid formation entitled genetic bottleneck. Much effort has been expended at gene pool enrichment through trait interrogression and interploidal hybridization in nearly every polyploid crop. In autopolyploids, transfer of genetic material between diploid and tetraploid levels is somewhat simplified by a single genome, a common cytoplasm, diploid gametes, and recognition of endosperm balance. These are my references.